Hey, it's Riffee here. I'm here to talk to you about Unit 2, and the particular part about Unit 2 I'm going to talk about within cells and tissue is how things get in and out of cells. Because, as you know from your patient needs IV fluid stat lab, that materials that are needed to survive or maintain homeostasis are actually absorbed at the cellular level and used within the cell, which then provide tissue to function, and we get into that whole interdependence thing. So we're going to talk about how things or nutrients or materials that are needed or waste that is not needed gets in and out of the cell membrane and how that functions. So, in order to understand how a cell functions and absorbs and gets things in and out, you have to understand the kind of the structure of the cell membrane. So, no matter what, all cells have inside of them cytoplasm, which is a fluid that suspends or allows the organelles and whatnot to float within it. But inside of that cell within the organelles and around that is actually a thin, flexible cell membrane, which can also be called the phospholipid bilayer. It's also called the plasma membrane, otherwise known as the cell membrane. The functions of that particular membrane can be broken down into two very general things. You can say that it regulates the substances that enter and exit the cell, and in some ways it kind of picks and chooses what goes in and out, and in other ways it actually can't help but let things go in and out. They move freely. The other thing it does is it provides support and protection. If you think about this, the inside of the cell is very delicate. That outside cell membrane actually provides a necessary or needed protection to allow the cell itself to maintain homeostasis, which then again leads to that whole interdependence thing where tissue can function properly, organs, then organ systems, and the whole organism works well as a whole. What you'll see here is you've got the picture of the phospholipid bilayer, and you'll notice that there's other things embedded in there, so it's not a misleading term, but a majority of the cell membrane is composed of phospholipids. Here you have the phospholipid heads, you have the tails, and you have the other end, which is hence the term bilayer, two layers of phospholipids, and their tails there. We're going to get into the characteristics of those here in just one second. Getting back into the structure, you will know that it's made of molecules, again called phospholipids, that are arranged in two layers, and that's why we call it a bilayer with the prefix bi, referring to two. The two major portions of any phospholipid is it has a head, which is the region of the phospholipid that is polar, and it also has a tail. These are the regions of the phospholipid that are nonpolar. This characteristic of polar and nonpolar really plays into effect in how the cell membrane actually sets up its structure and has the ability to remain in that set structure. When we talk about the phospholipids themselves in the bilayer, those polar heads are actually what are called hydrophilic heads. What that means is they enjoy water. They enjoy the interaction of water. They want to be next to water. And you have to think about that in terms of outside of the cell, or the extracellular fluid, which is the water or fluid outside of the cell, is largely composed of water. Also, inside the cell, that cytoplasm is largely composed of water itself. So that phospholipid bilayer, if the heads of those phospholipids actually enjoy being next to water, they can't help but want to be attracted to the water on both ends. Well, what that then does is it leaves the hydrophobic tails of the phospholipids on the inside layer. They do not enjoy water. They want to be away from it. So naturally, they try to get as far away. That then positions the heads toward the water, which like it, and the tails away from the water, which do not like it. Then that lends ourselves to this particular structure. So if there's water all outside the cell and water all inside the cell, and these heads enjoy being next to water and the tails don't, they like to get away from, as far away from that as possible, and the only thing they can do is form this bilayer structure. So, the plasma or cell membrane is composed of, I guess you could say millions and millions and millions of all these phospholipids. What they end up doing, though, however, is the phospholipids have the ability to move around within them. They don't have a set place or location within that cell membrane phospholipid bilayer structure. So what they actually have to do is they have the ability to move. Additionally, the other things that are plugged into that cell membrane, such as protein channels, carbohydrates, carrier proteins, receptor proteins, all kinds of different things, also have the ability to move within or in between those phospholipids. What this then becomes is what's called the fluid mosaic model. The fluid mosaic meaning it has movement and it has many pieces. That's what a mosaic is. You've seen these hanging in our cafeteria. Each person is assigned a square or a small piece of clay, and they put them all together to make one giant picture. But this is a fluid mosaic, meaning fluid, it has movement. We'll watch a quick video here real quick on fluid mosaic, courtesy of YouTube.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it.
So I want you to think, when they were disturbed to the point where they were out of place, those heads, they want to be next to the water, so they try to get next to the water. The tails want to be away, which naturally forms this head layer, tail layer on the inside, called the phospholipid bilayer.
Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside,
We'll get into these here in a
where they
second.
transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions.
Oh my god. Amazing.
The survival of all life rests on this veil of material. A supple
Interdependent.
membrane, just
the
two
yin-yang.
molecules.
So, let's get back into our notes. Absolutely riveting video. Here we go. So let's talk about cell membrane proteins. There are proteins embedded within that cell membrane. If you remember from the video we just watched, they have the ability to move around, which gives it that fluid mosaic model concept. But what they can do is they can act as channels or passageways for food molecules, other substances, so they can get into and out of the cell. What this then does is, based on what the cell needs or based on the concentrations of things in or out, with channel proteins, the cell membranes derives the concept of called being selectively permeable. Basically, using channel proteins, they act as gates or doors, and if the cell needs it, the door opens, things come in. If it doesn't need it, things go out. If it doesn't need it and everything's fine, the door remains closed and nothing can get in or out. The other thing cell membrane proteins do is they act as carriers by being able to selectively interact with specific molecules so they can cross. So some protein channels actually allow various substances through. Some of them act as carriers, meaning once a substance specific substance attaches directly to that, it will then carry it through the cell membrane to the inside or carry it from the inside of the cell membrane to out. That's different than just standard channels or passageways, which just generally allow things to go in or out. They can also act as receptors that transmit information to the cell by reacting with other molecules. So they can chemically react, and what that does is it generates a signal within the cell that kind of can tell the cell to do things. You can think of this like hormones. Hormones that travel throughout the body, such as adrenaline, can travel to certain cells within your body and actually trigger chemical reactions that then tell the cells to do specific things. You can kind of think of this maybe in lines of adrenaline and causing your heart rate to go faster during like a flight or fight syndrome. The other thing they do is they act as surface markers. Basically, there's just identifying proteins embedded in the cell membrane that kind of say, I am this kind of cell, or I am Matt Riffee's kind of cell. One of the big things you're going to study in this school year is actually blood type. Red blood cells have very specific membrane proteins on them that identify you as a certain blood type, either A or B or AB or O. A means you have only A proteins, B means you only have B proteins, AB means you have both, and blood type O means an absence of those proteins. And those identify those particular cells as one type. Here's just another example of the protein, or the phospholipid bilayer, or cell membrane. You have channel proteins, you have carrier proteins, you have protein molecules with a carbohydrate chain that could possibly be used as identification or carrier proteins. There's tons of different functions of those, and we're going to get into those in here in just a second. But before we do, I want you to take a second and pause these video notes and reflect upon the things that you actually have learned. And on an index card, which will be turned in 4 points, I want you to answer the following question. Question 1 of your entrance card, which will be turned in after you watch this video notes, is Based on what you have learned about the cell membrane, what properties of the cell membrane allow it to be selectively permeable? Or basically, what characteristics allow the cell membrane to pick and choose what goes in and out? Alright, I bet you got that question 100% correct. But I'm going to continue anyways. You can always rewind, pause, stop, do whatever you need to do. So let's continue with the cell membrane. The cell membrane is what is called selectively permeable, meaning it allows some things in, but not all, and some molecules pass through in and out freely. So here's what you have to think of. The cell membrane is kind of like the inside of the cell is where you live. So people want to protect themselves, similar to how a cell wants to protect themselves, so people will build themselves a house. So that house is built around you, keeps you safe, keeps you protected from the elements, and it also keeps you protected from zombies. So the house, being the cell membrane, can pick and choose what it wants to come in. Zombie knocks on your door, you don't have to open it because you don't need zombies within your house. But let's say your mom shows up, and your mom shows up and you kind of peek through the door and you kind of say, ooh, not right now, Mom, don't need you in there. She comes back later with cookies. You peek through the door, we need cookies inside the house. So you open the door, you let Mom in, you eat all the cookies, everything's hunky-dory. That's exactly how the cell membrane works. It has the ability to pick and choose what needs to go in and out of the cell. Small, non-charged molecules such as oxygen have the ability to flow freely through the cell membrane. They don't need any help. They move based on concentration gradients. Concentration gradients we'll get into in just a second. Small polar molecules such as carbon dioxide and water also have the ability to easily pass through following concentration gradient. Keep that in mind. Carbon dioxide and water, polar, can pass through easily. Oxygen, non-polar, but can also pass through easily. Then you have these things called macromolecules. Vaguely familiar from Unit 1. If you think about it, you have proteins, carbs, nucleic acids, and lipids, but they're larger molecules that don't have the ability to pass through the cell membrane freely. Example, glucose, one of the number one things that is required inside a cell, particularly the mitochondria, for use in cellular respiration to generate cellular energy, but does not have the ability to get in and out. But the cell needs it in. How does it get in? Well, we'll get into that in 1 second. But there's other certain molecules that are called ions. They're basically charged particles. The most common term that you hear these of are called electrolytes, but they have a difficulty crossing because of the hydrophilic or polar and nonpolar relationship of the phospholipid bilayer. And because of this, they have difficulty getting in on their own. They might not be big, but since their charge is the same as the hydrophilic portion of the phospholipid bilayer, they repel each other. Think of it like putting two magnets together. If you take two magnets and you put them together and the poles are the same on each side, you can't get those two magnets to stick together. You actually have to reverse the poles and have the opposites to get them in. That whole concept of opposites attract. So, molecules that have difficulty or need assistance crossing the cell membrane can get in a couple of ways. The first is called passive transport. Passive meaning if you're a very passive person, you don't exert much energy, you just kind of like, I don't care. Passive transport's kind of the same way. Basically, molecules move across the cell membrane without the use of cellular energy. Keep in mind, ATP is cellular energy. There are two types that we're going to go over for passive transport. You have diffusion and facilitated diffusion. Diffusion has a very specific one for water, which is called osmosis, but we'll get into that in a second. Diffusion and facilitated diffusion. Facilitate means to assist or help. So facilitated diffusion is a passive transport, but it requires some help from something without the use of cellular energy. The second way is called active transport. This actually uses ATP and it allows cellular energy. So there's something in the cell membrane that requires energy from within the cell to move particles, molecules, food, nutrients, whatever you want to call it, from outside to in, or also from inside to out. Both ways occur here. Some examples of active transport are phagocytosis, exocytosis, pinocytosis, and the sodium-potassium pump. The two that we're really going to get into in this particular school year are going to be phagocytosis and sodium-potassium pump. This one's huge later on, and you'll know all about it then. Let's talk about passive transport first. Remember, it does not require energy to get in and out. The first one we're going to talk about is diffusion. Diffusion is the random movement. It's really not random, but is the movement of molecules from an area of high concentration to an area of low concentration or less concentration. And concentration has, or solutions have two parts. They have something that is called a solvent, which is the thing that does the dissolving, and you have this thing called the solute, which is the object that gets dissolved. In humans, the ultimate solvent, or the thing that does all the dissolving in our body, is water. Think of it like this. You have a glass of water, you take some salt, you sprinkle that salt in there, the salt dissolves. But the thing is, the salt doesn't disappear, it just dissolves. So then you have salt water solution. The water is the solvent that does the dissolving, the salt is the solute. That's how your body works. So your water within your body, you've always heard that thing, oh, your body is made of 70% water, or whatever that number is. But that water, its main purpose is to dissolve materials so you can get stuff in and out of your cell. Concentration is how much water there is compared to how much stuff is being dissolved. The difference in concentration of a substance between the inside of the cell and the outside of the cell is called the concentration gradient. The ideal situation for things that pass through in diffusion is the inside of the cell and the outside of the cell are equal, and that concentration gradient does not exist, or it's in equilibrium. But there are occasions where the inside of the cell and the outside of the cell do not match. And then what happens is things that move through diffusion will always travel from an area of higher concentration to a low concentration. Actually, yes, that's true. Now, don't get this confused with osmosis. Okay, so let's look at it this way. Passive transport. High concentration of red dots, low concentration. If I were to draw a red line straight down the middle of this thing to indicate the cell membrane, whatever substance this is, if it moves by passive transport, particularly diffusion, it will naturally want to balance itself out from inside to outside or outside to inside, and it does so by moving molecules from a high concentration area to a low concentration area until they reach equilibrium. And even though they reach equilibrium, it's not like they each hang out on each side. They actually will evenly pass in and out routinely until, or keeping that equilibrium or balance. Simple diffusion is a really easy way for things like gases to get in and out of the cells. So you think of this like oxygen. This is how oxygen gets, you inhale. This is how oxygen enters your bloodstream, and how it also enters from your bloodstream into the actual cells of your body to be used in cellular respiration. If you want a real example, consider this little circle disc to be the cell. Keep in mind, intracellular fluid is the fluid inside the cell. Extracellular fluid is outside. If you notice, there's a higher concentration of red dots inside the cell and a lower concentration outside. Let's assume this object moves by passive transport diffusion. So naturally, it has this natural want to make everything equal outside to inside. So if there's a high concentration of red dots inside, those red dots are going to move out until both sides are equal. The opposite can occur too. There's a high concentration of red dots outside. They want to balance with inside. So the red dots are going to move from outside to inside until they're equal. And then red dots will move in and out freely but at equal amounts. That is called equilibrium. Okay. Osmosis is what we really, really want to talk about, because water is the substance of life. Remember, there's the whole thing, you can live for 2 weeks without food, but you can only live 3 days without water. So osmosis is a very special kind of diffusion that is the diffusion of only water. Water is the one thing that will move regularly to balance concentrations inside the cell and outside. So when water moves into a cell to balance concentrations, you get a greater increase of pressure inside the cell. And when it moves outside of the cell to balance, you get a decrease in pressure. That pressure involving water within and outside the cell is actually called osmotic pressure, and that actually can have drastic effects on the cell based on whether it is in a hypertonic or isotonic or hypotonic solution. Cytolysis is the opposite of when a cell swells. Cytolysis is when the concentration of a substance inside of a cell becomes so great that water will want to come in. So there's a really high concentration. We'll go back to the red dots thing. Let's say the red dots cannot move inside and outside the cell. So the only way to balance concentration is for water to move. So if there's a ton of red dots inside the cell, water is going to move in to try to balance the ratio of water to red dots. When that happens, the pressure inside becomes so great in that instances, it can become so great that the cell actually ruptures and bursts, and the cell will die. The opposite is called plasmolysis. This is when the concentration changes so much that water wants to leave the cell to balance, causing the pressure to decrease, and that cell will actually shrivel and wilt. This is actually what happens when plants wilt, but we're not going to talk about plants. We're talking about humans because this is human anatomy physiology. Water, therefore, moves just the same way from an area of high concentration to an area of low concentration of water. Keep that in mind. When osmosis occurs, water moves from an area of high concentration of water to an area of low concentration of water. You can look at it the opposite way. When there's a high concentration of something dissolved in water, that means there is less available water. So that means that in order to balance that out, it will want to get more water within. We'll get that in a second. A hypertonic solution is when you take a cell and you place it into a solution, and that solution that it has been placed in has a very, very high concentration of dissolved stuff and a very low concentration of water. In order for that to balance, water will move from inside the cell to outside the cell. The cell will shrink in size, and the outside of the cell will increase in water. A hypotonic solution is when you take a cell, you place it into a solution, and in that surrounding solution, there is more water outside than there is inside the cell. When that occurs, water will move from outside to in, causing the cell to swell, and if too much water enters, the cell will rupture and burst. The third type is an isotonic solution. Isotonic solution, basically, the inside and outside concentration of water and dissolved substances are equal and everything's hunky-dory. This is what we want. This is the ideal situation for when you receive IV fluids. IV fluids, you want to be in isotonic solution because you do not want the cells, while you're receiving an IV fluid, to want to take in too much water or release too much water, because that's a negative situation. In most situations when you're receiving an IV, you actually need that stuff. So you want it to be isotonic so your cells don't rupture, swell, or shrivel. Here's an example. So up at top here, what you're going to look at is this brown disc is supposed to represent a red blood cell. Red blood cells are placed into a solution in your body that is actually called plasma. They're all parts of your blood. This red blood cell here is placed in the blue solution, which would be the plasma. What you're going to look at here is, if you count the dots, there's pretty much an equal amount of dots inside to outside of the cell, which is an isotonic solution, and water will move inside and outside of the cell in equal amounts, and the cell shape is maintained and the function is maintained. In this particular example, what you'll notice is a hypotonic solution, which that will cause, and what these black dots represent on the outside are actually water molecules. When the concentration of water outside the cell is higher than inside, water will move in and cause the cell to shrink. Here's an actual picture of a red blood cell in a hypotonic solution, and it is swollen or swelled. Normal shape, they look kind of like a donut, but the hole, instead of being a hole, it's just caved in on each side in the middle. It's called a biconcave design. In a hypertonic solution, you'll notice there's less water on the outside and more on the inside, which water will then leave, which causes the shell to shrivel or shrink in size, and you end up with shriveled or shrunk red blood cells. These two situations get you away from homeostasis. This isotonic keeps you there, because if you remember, normal functioning internal environment. If you have your blood is hypotonic or hypertonic, you can get swelling or shrinking of red blood cells, which then causes you to not be able to carry oxygen and carbon dioxide effectively throughout the body, which then affects other cells, and you have this whole cascading series of events which ends up in you dying. Here's just another example of that particular situation they were talking about, and boom, entrance card question number 2. I'm going to let you read, I'm going to let you look at this, I'm going to sit here for a second and stare awkwardly into the camera. Feel free to pause it, answer the question on your index card, be ready to turn that in. Continuing on. Then we're going to get into facilitated diffusion. Facilitated diffusion still works on concentration gradients, but instead the substance that wants to get in or out based on the high concentration, the low concentration, that movement from high to low, it actually needs what are called carrier proteins. These are just proteins within the cell membrane that help the substance move across the cell membrane, but it does not use cellular energy. Keep that in mind. The result is still a movement from an area of high concentration to an area of low concentration. So you can just pick any random substance that actually moves through facilitated diffusion. If there's a lot of it outside the cell, it's going to want to get in. If there's a lot inside the cell, it's going to want to get out in order to balance those concentrations so the same amount inside and out. Here's a nice, absolutely positively beautiful picture of facilitated diffusion. So if you notice, there's a low concentration of 1, 2, 3, 4, 5, 6 of hexagons. Yeah, hexagons outside of the cell, or higher concentration outside compared to inside. This carrier protein is going to help or assist or facilitate the movement of this lipid insoluble solute into the cell cytoplasm. It does not use cellular energy, it just simply helps it move across because it can't move across freely. Now let's get into active transport. These actually move molecules. This is the movement of molecules with the use of cellular energy. And a lot of times it moves against the concentration gradient. So it can actually move from an area of low concentration to an area of high concentration. Or it can do the opposite. It can move from high to low. But regardless of what it does, it actually has to use cellular energy to get those things across. So whatever is embedded into the cell, the carrier protein or the sodium-potassium pump or whatever it may be, it actually requires ATP in order to function. When ATP runs out, it no longer functions. This is when you start getting into things like muscle cramps, muscle spasms, but we'll get into that when we get into those units. So you have various types of active transport. You have endocytosis. This is when a cell engulfs particles by actually extending out parts of their cell membrane, and it wraps around whatever it wants and creates a vesicle, which is a transport organelle within the cell, which once it encompasses itself around what it wants, the two pieces of cell membrane meet and they pinch off and it goes inside. There's two types of endocytosis. You have phagocytosis, which is the taking of large food particles or even substances that don't need to be there. You can think of this like as cell eating. It's kind of like the blob, where it kind of comes in contact with something and the whole blob kind of goes around it, and then whatever it comes in contact with is inside of it, and that's the end of the story. Then there's also pinocytosis. Phagocytosis with solid particles, pinocytosis is with solids or fluid. Both work in the same way, except for the fact that one's for solids and one's for liquids. Let's see if this video link works. And it does not. I apologize. Let's see if this one works. Oh, it's not deciding to work also. Fantastic. Well, I apologize. So here's just a fancy example. Let's say this is a food particle in this cell. It wants it. If you notice, it comes in contact with it. The cell membrane starts to pinch, starts to encompass itself around that food particle until the point where the two pieces of cell membrane touch and boom, pinches off. You have a vacuole. It carries it where it needs and gets absorbed. It's intense. Okay, pinocytosis, kind of soft. I don't know why. I like phagocytosis better, but here we go. So let's say that this is a dissolved material in the extracellular fluid and the cell needs it, but it's dissolved, so it's not a solid particle, it's dissolved within the fluid. So the cell membrane will instead engulf itself in that fluid, come in contact, pinch off, create a vesicle, be digested or used within the cell for whatever function it needs. Then you have exocytosis. This is taking stuff from inside the cell and spilling it out. There's a couple different ways. We're really going to get into this when we talk about secretions and excretions of certain substances within the body. But basically, it's the opposite of phagocytosis and exocytosis. So a substance instead will actually go from inside the cell in a vesicle, fuse with the cell membrane, and then it will be expelled out into the environment or extracellular fluid. Then you have sodium-potassium pump. I'm not going to dive into this hardcore, but it involves sodium and potassium and the use of cellular energy to transport sodium and potassium in and out, in and out, in and out within a cell. This is going to come into play when we talk about nerves and muscles. So I'm not going to get hardcore into this right now. But if you wanted to know, here's exocytosis. So this is something inside the cell. You can think of this as a hormone or some kind of product that the cells have made, and it needs to get out to travel to other places in the body. So whatever's made is put into a nice package, like a box, you could say, and then it's taken to the cell membrane, which you can consider this like, I guess, the post office. And then the post office will fuse that Golgi apparatus or vesicle with the cell membrane, and whatever's inside it flings out into the extracellular fluid, and bam, you have that traveling throughout the body. Now, we're into entrance card number 3, or question number 3. So eventually you're going to pause this video, you're going to take a minute, you're going to reflect about what you've learned, and here's what it is. There's going to be 2 videos on the next slide, 2 little small video screens that are going to show up in the slide, and what you're going to do, you're going to watch them. And what you have to decide is, by watching those videos, you have to pick which one is active transport and which one is passive transport by facilitated diffusion. Once you decide whether it's video A or video B, you need to actually label video A or video B as active transport or passive facilitated diffusion and justify your answer. Basically, explain to me how you know. Here's your riveting videos. Oh my god. Whew. Might not be able to sleep tonight. These videos are so exciting. Silly molecule. Oh, silly molecule. Are you seeing this? It's going in and out. It's crazy, right? You're welcome. If you have any questions over this stuff, you know what to do. See me in class. Send me an email. Shoot me a tweet, whatever you need to do to get a hold of me to answer questions. I'll be more than happy to help you out. I hope you enjoyed this as much as I did. Peace.